put my shirt in the focus here.
All right. Well, good afternoon, everyone. Welcome to the Living Earth Collaborative seminar. Today, we have the pleasure of listening to Dr. Renner talk to us. And yeah, I just want to, thank you for all coming. And so we're just going to get right into this. So we have the pleasure of actually having Dr. Renner now on our faculty here in the biology department at Washington University as an honorary professor of biology. With that being said, she has come a very long way to join us on our faculty here with many amazing positions and distinctions along the way. She actually originally got her PhD at University of Hamburg in Germany. And then she came over and had a position at the Smithsonian Institute in the US. And then she went over to Denmark, where she had a position, and I'm probably not going to say this name right. She tried to help me, and I, even though I am mostly Danish, I have no idea how to pronounce Danish, but it's the University of Aarhus, maybe something like that. I apologize to Denmark for not knowing how to pronounce that. And then after that, she went on to become a professor at the University of Mainz in Germany, and then became a professor at University of Missouri in St. Louis, where she then joined the faculty of the Missouri Botanical Garden, and then went back to Germany, where she was the chair professor of the University of Munich in biology, as well as the director of the Munich Botanical Garden and the Herbaria collection at the Botanical Garden in Munich, which is amazing, right? That is a lot. And then she finally decided that it was time to come back to St. Louis, where she's joined us for the last year or so. Of course, you don't get to have all those positions without being incredibly productive and brilliant scientists, which Dr. Renner has very much done. She has published over 300 journal articles, peer-reviewed publications, including several in the Proceedings of the National Academy of Science, Ecology Letters, and pretty much any other journal in the evolutionary ecology and systematics discipline. She has mentored tens of PhD students and tens of postdocs who have also gone on to do amazing things. However, I want to hear what she has to say. I could go on for another 10 to 15 minutes. We actually are kindred spirits on light pollution, and it's been an honor to start to work with Dr. Renner on fighting light pollution. But with that being said, let's hear what she has to say. Dr. Renner, it's all yours. Thank you.
Thank you so much. Thank you for this generous introduction, Brett. And it's so nice to be back in St. Louis and to be part of the Living Earth Collaborative, which is just really a wonderful thing. And what I want to talk about today is what is the best way to attach scientific names to a biological entity, and do we need new codes? Move forward. Okay. I suspect when evolutionary biologists hear there is going to be a talk about naming biological entities, their first thoughts are, we can never capture evolution in any consistent system of naming. If there is one thing we have learned from molecular data, it's how little overlap there often is between phenotypic traits that are easy to use to differentiate things and real genetic relationships. A prime example are the European crows, the hooded crow, Corvus coroni, described by Linnaeus, in the east, and the carrion crow, Corvus cornix, in the west. To the east and the west of a very narrow and stable hybrid zone, these species maintain their marked phenotypic differentiation and can easily be told apart. But genome-wide admixture analyses in 2014 in this paper showed that apart from one tiny region of their genome having to do with melanin production and the growing feathers, the northeastern German crows, the carrion crows, most closely resembled, or almost identical, 80% identical, the hooded crows from Poland rather than Spanish carrion crows. And you can see this in these two principal component analyses or axes overlaid with the map of the distribution of these two species. So in the commentary on this paper by Peter Neef, entitled How Carrion and Hooded Crows Defeat Linnaeus' Curse, he writes, ever since Linnaeus and his Systema Naturae invoked his taxonomic curse by requiring all biologists to use a stringent hierarchical species classification, researchers have been unable to define a single all-inclusive species concept. And these two crows provide a wonderful example of that, of the difficulty of putting biological entities into this rigid, hierarchical and captic system. And captic means box in box, meaning that one individual, any one individual can only be in one species, and any one species can be in one genus and never two. This is the box in box system. Such dimensionless boxes can clearly never capture evolution because they have no dimension, there is no time, there is no speciation, no hybridization, no extinction. This is dimensionless. Now, the first person to deal with how we might apply names to biological entities is, of course, Linnaeus. And he developed this hierarchic and captic system, which created many kinds of problems, including contributing to racism. And I am illustrating this here by showing you the page of Linnaeus' Systema Naturae 6th edition, in which he classified Homo sapiens, into these four varieties: the white Europeans, the red Americans, the yellow Asians, and the black Africans. And note the sequence in which he arranged these human varieties. A whole talk could be given about how Linnaeus elaborated this in further editions by adding more details and even a fifth category for monstrosities, which included anorexic girls. However, my talk today is not about criteria we use for circumscribing species, but instead it is about how we might apply names once we have decided we want to name something. And we might decide that we want to name a biological entity, which by definition is always incompletely known. We might decide we want to name it for two reasons. One is that we want to link our new entity to previous information in the literature, which is organized in databases or books or indices. And even if there's zero existing literature on our new entity, which we think is new, we want a short moniker by which we can communicate its traits or behaviors or endangered situation and need for conservation to others. And by placing this thing in a genus box, others can immediately extrapolate some of the likely features of the new entity, saving everybody a lot of time. For these two reasons, communication and extrapolation of traits from genus names, we need names. So how can we attach formal names as unambiguously as possible? This is the talk of my talk. This is the topic of my talk, which is about nomenclature, the rules of how we might do this. And as I already indicated, the first person to deal with the problem and solve the problem was Linnaeus. Here you see the itineraries of the 17 of his students that he sent out across the world to collect plants, but also animals, for example, a An elephant embryo, a pickled elephant embryo was sent to him in Uppsala. And of course, plants are just easier to send around. And this material was arriving from all over the world in no specific order. There might be, ascending from Cape Town one afternoon, and a couple of weeks later or days later, something would arrive from Venezuela, and something else from Northern Australia. And so he was receiving this stuff and having a wanting to do something with it. And here you see one of Linnaeus's special collection cases that he had built to order in his home in Hammarby. And you see that he used some of his early drafts here as wallpaper. And you see the naming of these pigeonholes. These are his orders. And there he placed the incoming material, regardless of whether it was from Venezuela or Australia, in the respective box. And here are two of Linnaeus's 900 index cards that have survived. Linnaeus appears to have been the first person we know that used index cards, which allowed to add things, okay? So here's an example of how he approached the problem. I want to stress that this pigeonholing is a key aspect of his approach. So here because of the season, of course, I use the pumpkin as an example here. We have Cucurbita pepo, which is the trivial name of the pumpkin. This is the trivial name. It says nothing other than that pepo, this thing here, belongs in the larger group denoted by this name, Cucurbita. This is today's species name. And the really important thing were these short diagnoses. In this case, it has lobed leaves and smooth fruits. Now, Linnaeus knew full well that there are hundreds of plants with lobed leaves and smooth fruits. But in this context here, the similar things may have lobed leaves, but verrucose fruits, or lobed leaves and longer stems and depressed fruits, or multiparted leaves. So in this context of similar things, lobed leaves and smooth fruits set the pumpkin apart. So this was his approach: a short diagnosis, a trivial name that was basically invented, citing earlier literature, mostly himself, citing illustrations when he had them. I will give an example later, and citing the native habitat when he knew it. For example, for the watermelon, he knew it from Sicily and Italy, but for the pumpkin, he didn't know the native habitat. He only knew it from cultivation. Now, these diagnoses are super important, and this is really another of his key inventions. And let me just read this paragraph from Stern's Botanical Latin. Linnaeus devoted much thought to the drafting of his diagnostic phrases. These are these phrases, which were for him the true names of species, and hence are very important for typifying, understanding Linnaean binomials. Linnaeus held that these diagnoses should not exceed 12 words in length, and he and Jacquin even managed on occasion to reduce them to one word. For example, Linnaeus's diagnosis of this plant from the southeastern US and Central America, this is, Eretia tinifolia. His diagnosis is Eretia enormis, the unarmed, the smooth, the non-hairy, the glabrous Eretia, which you can see here. And of course, there's another one that has hairs. So Linnaeus's invention of bipartite names, of which one indicates the outer box, that is the genus, within 15 years became adopted worldwide. It made possible a crowdsourced database. We really have to understand this. You couldn't upload your data, but you could certainly have a case built, a herbarium cabinet built, or an insect box where you would put your needled insects and arrange them in a system and communicate with each other. And so this was the way to name the world's biodiversity in a collaborative approach, enabled by the hierarchic and captic system. Now this works best when we have Linnaeus's specimens, a specimen, or duplicates of his specimens. When we don't have the specimen, as is the case for Drosophila, as I will explain, this can be problematic. So here's Linnaeus's diagnosis of Drosophila. It is, he called it Musa celeris, you know, the cellar fly, and he writes that it has such and such nerves in the wings and red eyes. And he also cites his earlier fauna, Swedish fauna, where he also gave the same diagnosis of this thing. And it lives in the wine cellars and similar places. Fair enough. But other people, you know, felt, in specifically this person, I forgot his first name, Meigen, felt that, you know, there could be confusion, that Linnaeus left no specimen, and really Drosophila should be newly named. And so Meigen had the audacity in 1830 to create a new name for Drosophila, Drosophila melanogaster, and place Linnaeus's name in synonymy, because he felt, well, there was no specimen, and, you know, we don't know what Linnaeus meant. And it's really amazing to me, because there can't be so many red tiny flies in Swedish wine cellars, you know, other than Drosophila melanogaster. But anyway, Meigen convinced the community that his name was the right one. He was associated with the specimen. And so today we have this name, and Linnaeus's nice name is a synonym. In addition, there are some 1,520 suspected synonyms of this name, Drosophila melanogaster. You can find them online. These are suspected synonyms, because I think without sequencing the genomes or part of the genomes of the types of all these 1,520 names, we really can't be sure. There might well be several hundred perfectly distinct species of Drosophila hidden among these names. Who knows? Conversely, there can also be problems when we do have Linnaeus' specimens. This happened with the watermelon. Here's the holotype of the name for the watermelon, Citrullus annatus, collected by one of Linnaeus' students near Cape Town. And when we sequenced a little bit of, you know, mitochondrial, nuclear, and plastid DNA from this holotype, it turned out that this is not what we now think of the watermelon. Instead, it's some other South African related species. So, as I've illustrated by these two examples, Drosophila melanogaster and the watermelon, names in our current system of naming are fixed to single specimens. Now, every evolutionary biologist in the audience will think, hey, what about variation? What about variation? Well, an attempt to cover variation are descriptions. Descriptions were invented by de Candolle, Alphonse de Candolle, around 1800. Here you see one of his descriptions. Up here is the trivial name. There is the diagnosis, and all of this stuff is description. Okay, all sorts. It has a root, it has a stem, it has leaves, it has a peduncle, it has flowers, it lacks a calyx, etc. pp. And clearly the idea is to have several specimens and then describe, you know, what you see there, perhaps covering 5 specimens or 500 specimens. The problem is that such texts include traits found in pretty much any plant, like a root, a stem, of many plants, and also hidden in here, something that is unique to this particular species. And so this, and of course, Linnaeus never faced this problem of variation. Linnaeus only had one specimen per species, essentially. For Swedish plants, he had sometimes more specimens. Even this, you know, this, I wanted to highlight this absence of trait. You can, of course, have any number of things that are lacking in your species of interest. Okay, this opens up a whole can of worms here. And species descriptions became longer and longer and longer, and really, in my opinion, useless, almost useless, certainly of limited use, the longer they are. And so if we look in the codes, that is the set of rules of how new species should be named, and we type into the International Code of Zoological Nomenclature, this is online, of course, we type into the search field description. Let's see what instructions we get. And so it says here, to be available, every new name published after 1930 must satisfy article so-and-so, must be accompanied by a description or definition. definition is the zoological word for diagnosis. Okay, description or diagnosis that states in words the characters that are purported to differentiate the new species from the previous one, such as leaves, lobed, smooth fruits. Okay, the description is voluntary. You can just have 2 words of definition. Let's see what the botanists have to say. So we go to the Botanical Code and we type into the search field description. And what do we find? Every new name must be accompanied by a Latin description or diagnosis. Now, this Latin description requirement was only the rule between 1935 and 2011. You can now do, since January 2011, English. Description or diagnosis. Description or diagnosis. Descriptions are voluntary. You don't have to describe your thing. You can just have a diagnosis. Now, the sharpest possible diagnosis, of course, comes from DNA, from nucleotides, where you could specify such and such a mutation sets your new species apart. And this has been done for now 21 years. Here's one early example from the year 2000 of a salamander. And this is based on, this salamander has 3 nucleotide substitutions in cytochrome B at position so-and-so and, whatever. And it also has morphological differentiation here from other related salamanders. But even if they had just given this diagnosis, this name would have been perfectly available. Here's another example for this little gastropod. This gastropod has reduced characters, whatever, and it is differentiated by a unique combination of nucleotides and cytochrome oxidase 1, barcoding, blah, blah, blah. And these differences are from the related species are shown in this picture. Here's another example. This is a plant we described. It has a molecular diagnosis in the NDHF gene, position so-and-so, has guanine, not thymine, etc., pp. Okay. Here's another one, a New Zealand centipede. This has an adenine in position 260 after primer 1F in the 18S ribosomal RNA, and this and that and the other. It also lacks spines and so on. Okay, this one, I love this one. This is a Nudibranch. This Nudibranch can be distinguished by 70 molecular diagnostic characters and cytochrome oxidase 1, and by the presence of a longitudinal medial silver stripe on the sole of the foot. You see this if you have a color photo of this organism. You can also use that, but I bet that the sequence deposited in GenBank will be much more used than this silvery stripe. Here are these marine worms. Okay, this is another example. And lest you think it's only odd marine things that are diagnosed with DNA, I put in these African turtles. These turtles, they have molecular, here, a gap of adenine, cytosine, thymine in position, blah, blah, blah. Okay, and also morphological traits. Okay. When I reviewed this topic in 2016, in this paper, I returned to Linnaeus's focus on diagnosis, not description, the use of DNA characters in the formal naming of species. I found about 100 examples like 80, fewer by 2014, by 2016. And today there are many hundred examples from all over the tree of life where people have used DNA-based characters to differentiate, to diagnose new species. And no change in any nomenclatural code was needed to do this because of Linnaeus's approach that a short diagnosis is all that is needed and a specimen. Okay. So to summarize to this point, Linnaeus's approach for attaching names to biological entities always incompletely known consists in having a specimen, attaching a name to it, and publishing a sharp diagnosis and citing images and geography when available. If he could speak now, he would certainly say, and he would certainly say we should add recommendations in the codes to cite images and DNA-based diagnoses where feasible. And so following on Linnaeus's suggestion, I have in fact submitted it's already published, a recommendation for adding photographs of type specimens to the Protologs. Protolog is a technical term for first, where you first publish a new name of the names of taxa. Now this is actually a trivial proposal. I do think it will be useful to have photographs of types if you think what computer vision and machine learning can do with images and matching of images. I do think this will be useful. But I'm putting in this harmless proposal to lead me to the second and final part of my talk, which is about how are the codes improved? How do the communities of people who are interested in naming improve our ability to name things? And what is the current hot topic? So do we need new codes, and how are codes changed? Now the botanists and zoologists and other taxonomic communities do this quite different from each other. And so let me go through the 5 main codes and how people change their codes. Okay, the currently permitted name-bearing material in the 5 main codes. Okay, there's the zoological code, the code for algae and plants, the code for fungi, which has been separated from the plant code since 2018, the codes for Prokaryote, and then there is a new draft code that is being finalized as we speak for uncultivated prokaryotes. So let me start with the Micrological code. In the Micrological code, for you to correctly publish a new name, you have to have either a dried mushroom, a specimen, a dried mushroom, or an inactivated, dead culture of hyphae. Or you can use an illustration. DNA is not permitted. A DNA is not permitted, a genome, for example, is not permitted to be the name-bearing material. You have to have a specimen, an inactivated culture, or a picture, which may strike many of us as crazy. So the Micrological code that I'm talking about right now covers fungi, or mycota, and other unrelated fungus-like organisms, many of which are of great medical importance. The world's culture collections currently hold strains of some 30,000 fungal species. That is about 3% of the suspected 1 million unnamed fungi in environmental samples. Since 2012, the internal transcribed spacer region of ribosomal DNA is the universal barcode for fungi. And there is an obligatory registration of new names where you have to cite that specimen or the dead culture or the illustration, and usually you will also cite a barcode. Now, if we look at this picture here from Nature Microbiology this year, May this year, it's just really a great illustration. This is the environmental samples of this barcoding region generated from the fungal microbiome of switchgrass at one site in North Carolina. And each dot is a different ITS sequence, okay? Cluster A, Basidium mycota, Ascomycota, these things, and cluster B. And so the Micrological, and none of them are cultivated, none of them are dried, available as dried or dead culture. So the Micrological community is currently at an impasse, as beautifully discussed in this multi-author paper. Okay, as I explained, under the current code, you have to have a dried mushroom or inactivated culture. One way out would be to focus more on the other option, which is an image. So you could have a fluorescence in situ hybridization image, but it's a lot of work to do these images. And I'm personally, I doubt this is a useful approach. Or you could change the code, the Micrological code, and allow DNA sequences as type, as name-bearing material. And presumably this would help name this, cataloging, or would help speed the naming of these so-called dark taxa that we just saw. Here they are, dark. They haven't decided, and it's a very acrimonious debate. Now let's move to the prokaryote community. The Prokaryote code, the nomenclature code for prokaryotes stipulates, get this, that for you to validly publish a new species of prokaryote, you have to have cultivated living strains. And the code says viable cultures must be deposited in at least 2 publicly accessible culture collections in different countries from which subcultures must be available. Now this incredibly detailed and, in my opinion, dubious rule has made things extremely difficult, because you have to cultivate the stuff, this prokaryote, and then you have to send it to 2 public cultures, which of course are all in the developed world, because who can pay for these things? It's extremely expensive to maintain these cultures, and of course they mutate and they get contaminated, and then some culture, some collection gets defunded, so the stuff is no longer as available to be sent around to researchers who might be interested. So this rule has really created problems for the micrological community, prokaryote community. Illustrations are not permitted, and DNA sequences, amazingly, are not permitted. And of course, this has led to a split in the community. Here's an image. These are the public repositories, currently contain 70,000 bacterial and archaeal genomes. And in this paper, 8,000 of these 70,000 have been analyzed, high quality ones. And you can see that in this phylogenetic tree. All the ones that are in green, which are 17 phyla in this tree, comprise uncultivated bacteria and archaea, which will never be named as long as we have the current code for the naming of prokaryotes. And this situation has led Barney Whitman, who for the past, I think, 30 years has really almost single-handedly updated and authored or updated this manual of determinative bacteriology, to publish what he called, this wonderful understatement, modest proposal to expand the type material for naming prokaryotes. Okay, the code for prokaryotes should be modified to allow gene sequences to serve as the type material for genera. It's very interesting, not for species, for genera in the absence of described species. Now this was met with huge interest, and NSF has funded 2 workshops earlier this year. I was invited to attend them. It was super exciting, where people, the prokaryote experts, are currently writing a new code, which they call the sequence code, and which they hope will become adopted by the community. We are meeting on a regular basis so that the nomenclatural framework for the DNA-based type material, cultivated and uncultivated, of course, they hope to get everybody under one code, becomes a fact soon, and researchers can start naming accordingly. Okay, so let's move on to the zoological code. The zoological code, the current code, permits specimens, of course, illustrations, because Linnaeus also used illustrations, and they have found a way to actually use DNA sequences in a super interesting so-called rule that they call Declaration 45. So the zoological code is updated in a different way than the botanists do it. They have a standing commission of men, in this case, that meet a couple times a year and then vote on any proposed changes. And so these people voted in December 2016, 21 in favor, 3 against, 3 abstentions on this Declaration 45. And it concerns specimens preserved. What does that mean? I'm explaining this on my next slide. Okay? I'm not going to explain it. The president of the Commission for the Zoological Code will explain it. He sent me this wonderful, very clear email explaining Declaration 45. The Zoological Code still requires that a new species has a physical name-bearing type, but this type could in principle have disappeared, disintegrated, liberated into nature or something similar. The recent fire at the Museo Nacional in Rio did not invalidate new species in press or in prep, for which the holotypes went up in flames. Our Declaration 45 also means that a holotype that was entirely destroyed during DNA extraction would still count as the name-bearing type, although the only evidence of that specimen would be the nucleotide sequences. Therefore, although the sequences by themselves cannot be types, they would be de facto types. I just love it. And you can imagine how this is so relevant, because any new mammal, such as these 10 new mammals described in the 10 species of mammals described in the 10 new years, it would be unethical to kill them unless you found a dead one. You would have, the DNA from the poop or the hairs, you would have video of the behavior, you would have sounds, you would have everything, and you wouldn't want to kill this thing unless it were, there's really no need. Unless you are interested in the liver or something, but you might not get permission to kill it, because by definition, any mammal that is described now is going to be rare. Okay, and this other thing about the holotype having been used up in DNA isolation is also not a crazy situation. This has in fact happened several times already. Here are some tiny gastropods, and the authors write, of this new species, write, due to the minute size of the animals, entire specimens were used for extraction. Consequently, the holotype is a voucher of extracted DNA. This is only possible because of the Declaration 45, which covers this situation when the holotype has disappeared or been used up or been liberated into nature. Here's another one. Here's a new species of hyrax described just very recently. Here's the skin, the skull, and they have in fact a specimen. Okay, they have a wonderful movie of the thing, and they cite these sequences. And should that specimen go up in flames, the DNA sequences would serve as the de facto type. Now, what do the botanists do? The botanists, of course, are conservative. We always are. So the code for algae and plants permits specimens and illustrations, but no DNA as name-bearing material. Now, algae, which are covered under our code, consists of numerous unrelated branches in the tree of life. All these names that are in brown or green or turquoise boxes are covered by algae. Cyanobacteria, brown algae, the green algae, and all sorts of other, the dinoflagellates. These are all algae covered by our code. Now, when Linnaeus had to deal with algae, for example, when he wanted to name Volvox globata, he relied on drawings. He studied Volvox globata under his microscope. People often don't realize that Linnaeus loved microscopy. This is his actual microscope. And so he cited for this thing a drawing and another drawing. And then he says where it lived. Okay? And today, when we describe a new algae, just as this one here, here's the holotype of this new thing. And there is in the publication a cited sequence. Now, under the Botanical Code, this sequence has no relevance. It's just, we can't use it as type material. It's interesting to have it. But under the Zoological Code with their Declaration 45, this could serve as the de facto type if this specimen were destroyed, or you have no access to it, or you don't want to look at it under a microscope because really you have no time. Many organisms that are covered under, that are part of algae in the right sense, are covered or treated under several codes. For example, dinoflagellates and dinophyceae. When they have chlorophyll, they are plants, and when they don't have chlorophyll, they are animals. And this occurs sometimes in different stages of life of the same species, and certainly in various species of these things. So some of them are treated under the Zoological Code, and some are under the Botanical Code. The same is true for cyanobacteria. Some of them are treated under the Prokaryote Code, and some under the Botanical Code, okay, such as no-stop. There are many examples. So being the optimist that I am, I did submit a proposal. It is now in press. I submitted it on the 28th of September. A proposal to permit nuclear DNA sequences as nomenclatural types when the preservation of specimens is not feasible. Now, I specified, all the conditions, and And I set as the starting date, I proposed as the starting date, January 2026. After this date, the type of a new name of algae, I'm thinking of algae really, might be an assembled nuclear genome or informative part thereof. Now this optimistic proposal of mine will be voted on in the nomenclature session in Madrid in June 2024. And who goes to the nomenclature session, or the only person allowed to vote and people allowed to vote in the nomenclature session are curators working at recognized herbaria. The big herbaria get several votes and the small herbaria have single votes. And you can imagine that, of course, the people who will be voting on such proposals, many other proposals, are by definition not working on genomics. And they almost certainly will vote down this proposal, but you have to start somewhere. And I want to end on a positive note. The codes of nomenclature, all the 5 codes that I've been discussing, there are 2 more. There's a code for cultivated plants and there is a code for virus naming. Okay, so these codes of nomenclature are an instance of 170 years of peaceful international collaboration, despite umpteen wars. The first code, which was a zoological code, was written in 1843, and one of the people on the committee who developed this code was Darwin. And thinking about Darwin and nomenclature reminded me of a letter I sent to Science almost 30 years ago. The best thing about this letter, here's my letter. The best thing about this letter is that it was published next to a letter by Ernst Mayr. We both wrote about a previous paper called Ordering Systems. Okay, and at the end of my letter, I pointed out that Linnaeus may even have had some influence on the development of modern ideas on evolution through his work on the economy of nature. This work, the Economia Naturae, which discusses the warfare, competition between species, was studied by Darwin. And Darwin studied it and other publications by Linnaeus because he was on this committee developing the first code of nomenclature for animals, which was essentially copied from Linnaeus' suggestions. So Darwin read Linnaeus, and while Homo sapiens heads the table in Systema Naturae, as I showed you in my second slide, he is immediately followed by the monkey. Here we are, back at Homo, nosce te ipsum, know thyself, and here's the monkey. Thank you very much.
Excellent talk, Dr. Renner. It's fantastic. I can hear all of St. Louis applauding as we speak. Man, I really liked your talk. Very thought-provoking. And I have to say, right, I'm a zoologist and I take for granted all the naming that has come before me that allows me to just go out into the forest and collect specimens. So I have to ask, this will be further discussion that you now have, but, you know, of course, the biology is quite different between eukaryotes and prokaryotes. And so does it even make sense to have a similar naming system between the two? And especially like with what little I know about bacteria, right? Like, how do you even name that? Because it's really just clones, right?
Yes,
so how do you even do it? How does this work?
Yes, that's the way it's going. More and more and more codes. I mean, as I showed in my table, there are 4 main codes, and the prokaryotes, people are just writing a new code as we speak. And that's the direction it's going. Okay?
Yeah.
The disadvantage is that there are these deep lineages in the tree of life, like dinoflagellates and cyanobacteria, that are now treated under 2 codes and potentially actually already under 3 codes, because there will be 2 prokaryote codes very soon. So that's the danger.
Yes, it seems very messy. And then the other thing is how, this whole idea of relating a specific species to a specific sample, yes, that works if you're short-sighted, but how long can a plant sample actually exist, right? I mean, like 1,000 years, it will degrade, right? I mean, no matter what we do, eventually science is going to lose those samples. So what do we do about that too?
Yes, that also certainly applies to DNA sequences, because, you know, if by some terrorist attack and the CBI gets attacked, the server crashes, whatever. Yes, yeah, I don't know. If we have multiple sources of information, you know, literature and images and sequences and specimens, you know, hopefully we will have some rest left where we can say, this is what Linnaeus meant, or what Susanne Renner meant in the year 2021. It is a problem. It is definitely a problem. But it's just, I mean, this problem of preservation of material is independent of attaching names, yeah?
Yeah, absolutely. Excellent. Well, actually, Liz, do you want to take the next one?
Yeah, so we have a question from Dr. Losa. I'm curious about your thoughts on the Code and in general, whether it has implications for your proposal.
Yes. Oh, but Jonathan, I love the Code, and I've been teaching, as when I was teaching every year, I taught this. It's wonderful for discussion. The Code is not relevant to what we are talking about here, which is attaching names to species. And as you may know, the Code does not cover species. You would apply labels, you would stick labels to single specimens, and those specimens are the type of the name so-and-so, as in the current codes.
So I have a question personally kind of on that realm, but one of the problems that I have inherently with species names and species delineations is that evolution is this ongoing process. Yes. And so kind of how Brett said, well, how long can we keep these samples? How do we deal with the fact that if we're seeing rapid evolution and rapid speciation, what do we do when 1,000 years down the line?
I wouldn't worry about it. I wouldn't worry about it. The nomenclature is about attaching a name to one specimen, okay? It is not about capturing evolution, as I tried to make clear in the beginning. The topic I was talking about is unambiguously attaching a name to one specimen. And then the other people are free to add another 5,000 Drosophila melanogastas to that name, or instead describe 500 new ones. Up to you. There is no rule. The nomenclature doesn't address the question of species circumscription. It doesn't address it. It's a system of rules of how to attach a name to a specimen. It is so difficult to understand, and it's so important to understand that descriptions are voluntary. It's words, they don't matter. If the description says red and the specimen is blue what counts.
Yeah, that's a really good way of putting it.
And it's because Linnaeus didn't rely on descriptions, and he didn't have to cover, or he couldn't cover variation because he didn't see variation. How could he have?
Right. So Dr. Jones-Strassman has a really great point here, and I'd love to hear your comment on this. So she says, my group named 3 bacteria species, and it was a huge amount of work to get them through, but we had the sequences. There are more bacteria we cannot name because they cannot live independently, right? So there are bacteria how are you going to culture them? Right.
Yeah, you can't culture them. I only understood this when I attended this, and it was a 2 full-day workshop NSF. I invited only maybe 50 people. And I really only understood that there are tens of thousands of important or interesting, maybe not tens of thousands, but there are certainly tens of thousands of bacteria that cannot be cultivated. And some of them are very interesting. And they cannot be named under the current code. And then you have to have 2 cultures in 2 countries, etc. I mean, it's crazy.
Yeah, that is insane and incredibly non-inclusive.
Oh, but it was for high quality. They wanted high quality. These people are obsessed. I only understood that when I attended this workshop. The bacteriological community is obsessed with quality control. They want you to register the name and check everything and whatnot. In botany, there is no quality control. None. You know, you name this thing, you put it in the herbarium, you say, this is my new species. And the quality control, it lies only in whether the community adopts your name.
So Dr. Axelrod has a question here. What's the opposition for allowing sequence data as a basis for nomenclature?
Yeah, I did not represent this opposition. The opposition is very strong. I've given similar shorter talks in communities, addressed to herbarium, to people working in herbaria and zoological collections. And the opposition is huge because they feel, that people working in collections feel that it would somehow reduce the importance of collections, which of course it wouldn't, because the whole thing is voluntary. Where you face the situation that you can't make a good specimen, as Jones Strassman with her bacteria, okay? Then you use the DNA. If you can easily make a specimen, you make a specimen, put it in the herbarium or the zoological collection. But they feel that it somehow devalues, that's my feeling. It's very, very strong opposition. I mean, an excellent, thoughtful people hate the idea.
All right, well, I think we have run through all of the questions. And again, I have many more things I'd like to talk to you about, but this was fantastic. Just really great to think through. When I first saw your title, I thought this is going to be really interesting. Like, I don't know how we can talk about this for an hour. And it is just, I'm only going to be thinking about this for weeks, like how it is such a mess. And of course, there's the whole spectrum of species and everything. But this was really thought provoking. And thanks so much for giving us a talk.
Thank you.
And everyone, have a great fall break, and we'll see you back next Thursday for our next talk.
Thank you.